Good. Hello, hello. We are probably, there will be more people coming, but I guess we could already start and welcome everyone. So thanks a lot for signing up for the webinar. And we start with a few slides introducing the Bioimage Informatics facility, so us, and then basically Christoph will take over and present TissUUmaps, for what you are here for. So let me share my screen. And then, do you see that well, I hope? Good. Yeah, thanks a lot. So, I mean, I hope you kind of people from Sweden already know us. So we are kind of a national facility offering bioimage analysis support. And then you can see that we are located in both Uppsala and in KTH in Solna. And basically, we can really provide you with a lot of like support and training on image analysis. We provide like machine learning tools, of course, bioinformatics, computer vision. So if you have the main message here is if you have questions about image analysis in your projects and you are located in Sweden, then you can easily contact us over this email address. And then here today is, so you will see Christoph and Fredrik mainly presenting this. And then we have also Jonas here already, who will join us in April. And Giselle is maybe here, she is a bit busy. She says hello. And so what we are doing is, I mean, we have, of course, since we are a national facility, it can be really a broad range of projects that we are working on. Here, just picked like three that are a bit more in spatial omics, because I guess you might come more from that side. So we have quite some experience by now in this multiplex tissue analysis with like how to segment cells and classify them and how to do spatial analysis. And then also like in situ sequencing, spatial transcriptomics, and they are especially visualizing the data with the TissUUmaps. But that's not everything. So we can really, I mean, if you have some spheroids or some even more image data that is not related to spatial omics, then we can also handle that probably and just let us know. And that we have also this project database that you can look a bit more on the projects we have been handling. And that's already kind of the bridge to Christoph and Fredrik. So it's they will present, or I think mainly Christoph who's presenting, and Fredrik is helping in the chat and so on. And so I give the stage to Christoph presenting TissUUmaps.
Thank you, Anna. Very nice introduction. Let me share my screen. Can everybody see the screen?
Yes.
Perfect. Okay, so yes, today I wanted to talk a bit about TissUUmap 3 and more especially about TissUUmap 3.1 that was released a couple of weeks ago. But as everybody doesn't know about TissUUmaps, I will start with a small introduction about what TissUUmaps is, where it comes from, and what you can do today. And then I will move on to the a bit more specialized new features of the last version. So, can I have focus? Yes. So everything started for TissUUmaps in 2016 as an ERC project. It was actually called TissUUmaps with a normal tissue in the beginning, and was renamed in 2018 as TissUUmaps for kind of copyright reasons. But in 2016, the project started to explore spatial and genetic information with Carolina Velby's lab. And the idea was really to have a pyramidal view of the images and also gene expression on top of it, and to have that as a web-based interface. In 2018, the development of TissUUmaps started really, and we had a first version developed by mainly Leslie Solorzano, and Gabriele Partel, and you can find the first publication about TissUUmaps in the bottom. And the idea here was to have a web page where you could load static images. So you had to convert your images into a special format to display. And then you could like display CSV markers on top, and everything was in SVG format. So it was pretty slow, but already looking pretty much like TissUUmaps that we have today. If you look at the capture on the right, it's very similar to what we can use today. Then in 2020, we started to have a background server that was running in Python with Flask, so that you can actually generate all your images on the flight, and you don't have to create the DCI file anymore. And also we moved to WebGL for the markers, so that you can now display millions of markers instead of thousands, and with no extra loading time. So it's way more powerful to display, for example, in situ sequencing data, where you have up to 10 million markers on one sample. Then in 2021, we released TissUUmaps 3 after a big refactoring of the whole code and adding plugins and Jupyter integration, so that you can run TissUUmaps in the notebook, and also deploy everything as a Docker server, so that if you have, for example, a Kubernetes instance, you can deploy TissUUmaps on it and have many collaborators working on the same instance and share the data. And that was 2 years ago. And then this year, or 2020, 2023, we released TissUUmaps 3.1. And now we can load AnnData files directly. We can have multiple images in parallel, so that you can compare samples or compare different layers directly next to each other in TissUUmaps. And also network diagrams visualization. For example, if you're working with QuickPy, you can have like neighborhood networks, and you can display that on the spatial image, or in a feature space, or any kind of networks that you would have. And that's what I want to talk a bit more today. But first, just to show you what TissUUmaps looks like. It's an interactive viewer. You have the web version, so you can like browse in your usual browser like Chrome or Edge or any other modern browser. And you also have a desktop viewer. So if you want to install locally, you can install the graphical user interface and have TissUUmaps running on your computer, load your images, load your markers, load your regions, and then play with that locally before you share it with others. But the main focus is really that everything should be available as a web page. And then since TissUUmaps 3, we also have Jupyter Notebook integration. So if you have images, markers, and data files, any kind of things you want to display in your notebook, you can directly call the TissUUmaps Jupyter functions and display it in the notebook and export it to a web page also. For all that, you can install locally with pip in Python, or you have a specific Windows installer if you want the graphical user interface, or you can put it as a Docker container. And we have our webpage, tissueumaps.github.io, if you want more details about the installation or usage. We have documentation on it also, if you want to see how to use the different features. We have mainly three kind of data that we can display in TissUUmaps. We have images. For now, it's mainly like 2D images. We are working on a new version that could be able to display multi-dimensional images directly also, but it's still work in progress. So you need to have your images as 8-bit because that's what the browser can display. If they are not 8-bit, then it will be converted automatically before viewing. So be careful with your range and everything. It's better to pre-process your images so that they're already 8-bit and 2D. But yeah, everything will be open, but you can have surprises if it's not 8-bit. We can have regions that are exported from other tools like QPath or Cellpose or Stardist or any segmentation models or like other, or Napari. We can load in regions as GeoJSON or directly path objects. And then we can interact with the regions. We can export the list of markers inside each region and things like that. We can also draw new regions manually. And then we have the markers. And here we worked on having a way to load as much markers as possible. So right now we can load up to 10 million markers on one sample. And they have like X and Y coordinates, and then you can display different colors, shape, size, opacity. You can also display pie charts if you want. So if you have multiple values for each like cell type, probability, or things like that, you can display markers on top of your like pie charts instead of your markers. And more recently, we can also load directly AnnData objects. So it's annotated data from the scverse package. So from ScanPy, SqueezePy, and everything. And yes, so I will show you a bit how we can work with these, but basically in one AnnData, you can have images, you can have markers, you can have many more of these kind of things directly in one file. So just to start with the 2D images, you can have many 2D images, and you can go through them one by one like that, and display like each in different colors if you want, or you can display them next to each other, and you have some features that you can apply to your images. You have a list of filters, you have like, you can play with gamma, you can play with the opacity, saturation, brightness, you can do all that directly on each image separately. Then you have the markers, and here we can display 2 million markers, for example, on top of the data, and each marker has a specific color for one targeted gene in this example, and a specific shape. You could also have specific like pie charts or different size, depending on what you want to display. We try to make TissUUmaps as open to what you want to show as possible, so it's very flexible in the settings you apply. That's an example of pie charts. So you can see you have the legend on the left. Here we have many different different sectors, so it's not easy to read. But if you have like 5 or 6, it's the legend would be a bit easier. But here you can see for each pie chart the cell type probability, and then you can like select the ones that you want, and so on. And finally, how to draw regions on top of your images. So either you have the like click and draw regions like that if you want to highlight a specific region of interest. And then you can export them to GeoJSON and load them into QPath or CellPose or anything. Or you can import GeoJSON from other tools. And here we will import the cluster regions, and then directly you have all your regions on top of the tissue, and you can like fill them and see what's in it in each layer. So try to make TissUUmaps compatible to all other softwares. I see that we have some question is in the chat. So yes, let me know if there is any question that you want me to answer, but I see that Frederick and Jonas are working on that. So thank you, but don't hesitate to ask also here. Okay, and finally, the AnnData objects, which is annotated data, and that can contain images and many different kind of observations. So for example, here we have data coming from in situ sequencing techniques. So we have many spots, and for each spot, we have a number of counts. We have many different like quality control things we can display here. So if we click on total counts, we will directly see on the left the total counts for the different samples in the AnnData object. I will come back to that, but you can see here that we have 3 samples in parallel, so we can display as many samples as we want. AnnData comes with library ID, so you can have many different images, and the markers will be associated with each image separately. And to finish on TissUUmaps' 3 main features, we try to make it as possible as compatible as possible with other existing softwares. So here we see, like, if you export things from Python, you can load them in TissUUmaps. You can also interact with like usual image analysis softwares, or with like ScanPy and other tools like that.
So there's a question on the chat that maybe relates to this, which is, are you planning on integrating OMISAR as a supported format?
That's a very good question. So it's we are thinking about it. The problem is OMISAR is evolving quite fast. It's not completely like, yeah, there is a lot of work on like NGFF and other kind of formats to incorporate more objects into one format. And we are not sure we can follow right now. So we focused on AnnData, but if people want to work on integration with more formats, or if OMISAR or NGFF formats are a bit more widely used in the future, we might incorporate them also. But yeah, right now, TissUUmaps is a research project and we don't really have the time to like make it compatible with all formats. Does that answer the question? I'm sorry, I'm a bit sick today, so I might cough a bit. Okay. And then I wanted to discuss also about plugins and show you some examples of the plugins we have already in TissUUmaps. So like the feature space plugins, or converting in-situ sequencing points into region clustered, or doing quality control on in-situ sequencing. So here is a feature space plugin. Basically, if you have spatial data on the left and you have also for each point some UMAP or GSNE or PCA or any other kind of feature space data coordinates, you can display them next to each other with this plugin. And you can interact with the left side and with the right side. And you can also display the histogram of what you selected and then see where they fit on each side. So this is very convenient if you want to understand your UMAP data, for example, or if you want to see where some cell types are in UMAP and in the data. The correlation, the coloration, and everything will be the same on the left and the right. Another way to display the feature space is in 3D. If you have like UMAP 1, 2, and 3, then you can display that. You can rotate it. It's a bit more difficult to interact with 3D. So right now it's only display, but maybe in the future we would have like more interactive way of showing also interaction in 3D. That's a plugin that was developed by Axel Andersson from Velby's lab, a PhD student. And the idea here is to have a very fast way to cluster data when you have many points of different classes, and you want to find regions that are similar in expression. So here, for example, you see that from in-situ sequencing data, we cluster the data into 4 regions, and it takes a couple of seconds to run. You can change the size of your neighborhood that you want to take into account, and so on. That's the kind of like plugins that you can develop. And we tried with TissUUmaps 3.1 to make the plugin API as simple as possible in JavaScript, so that you can create the right interface for your plugin as fast as possible. And the last plugin I wanted to talk about is Spot Inspector. And here the idea is that we have in-situ sequencing data, and that comes from many different images on top of each other, because you have multiple rounds, and for each round you have multiple channels. In this example, we have 5 rounds and 4 channels, so a total of 20 images. And then these rounds and channels are decoded into markers. And we can see by clicking on the markers why it was decoded in that way by looking at the path that you can see in the plot on the right. And then you can do quality control on the decoding and decide if your decoding was correct or not, and why this gene was found and this one was not, and so on. So this is like a plugin working directly on the raw data, but with like interaction also with the marker lists. And we could also have plugins working with images, markers, and regions, and so on. Okay, so If you want to see a bit more about all these, you can check our video on TissUUmaps 3.0 that is on YouTube, actually a previous webinar of the bioinformatics facility. And here we go through all of these examples I showed and detail a bit more how to load them in TissUUmaps, how to play with them, how to change your settings, and so on. So yeah, if you have any doubts or you want to spend a bit more time, you can check that. And I see that we have a question on the chat about using it for your own single-cell RNA and ST-seq data. So right now, you can load any data you want, either locally on your computer or deploy it on your own server. As long as it has coordinates, you can have images on the background or not. I can show you some example with or without images. You can have as many points as you want, usually up to about 10 millions. After that, it starts to be slow to load. But yes, you can really load and deploy any data you have. The simpler, and I will start to talk about that now, is if you have it in AnnData format or in like HDF5 format, because then the integration is very fast. You can just like drag and drop your AnnData objects into TissUUmaps and it will display all the observations in it. And then you can export it to a static web page or put it on a Docker instance on the web if you have like a Kubernetes server or anything like that. I will show you that in the following. So to focus a bit more on TissUUmaps 3.1, what we added on this version, and I talked about that already, but it's the HDF5 format and AnnData support. I will show you an example of both. Actually, AnnData is written in HDF5, so we had to make the HDF5 support to read the AnnData. Also the collection mode, where you can display images side by side and have markers on each image separately. We also have network visualization, so graphs. If you have graphs on your data that comes from SquidPy or from anything else, then you can display the edges directly on your data. And also worked a bit on the plugin API. I will not talk about that today, but if you have questions about the plugin API, you can contact us. And we are working on adding that to the documentation also, so that it's simpler to use. So to go back to the marker formats and to HDF5 format, historically TissUUmaps could read data from CSV files only. The CSV file is comma-separated values, which means you have a big table of values into one file, and it's in text format. And in TissUUmaps, you had to load the entire file to display just the columns you want. So if you have like X, Y, cell type, quality, and other things, you need to load all the files. It's not efficient for loading time also, because it can be quite huge and then you have to wait until it's finished. And if you have, for example, spatial transcriptomics, where you have 3,000 different genes and you want to display one at a time, then you will have to have one CSV file per gene. So it's we were limited in how to create projects for this kind of visualization. So that's why we worked on having HDF5 files where you can have lazy operations. So you only load the data you need. You can slice the data. You can access any part of the data you want. And it's HDF5 means hierarchical data format. So it's really, you can really select the data you want. So it makes a way faster download. You don't have to download the whole file. And also, as it's like the format of AnnData, it's very good for integration with ScanPy, SquidPy, etc. I took these slides from the HDF website. So it's just to show you what is HDF5 really. So it's, like I said, a hierarchical data format. And basically it comes with a data model, a library, and a file format to manage your data. And you also have tools that they build to access the data and with different programming languages and so on. To summarize, HDF5, it's like a big box or your luggage in which you put all your objects that you want, and you can have many different kind of objects. To see that a bit more as a structure, you will have your root object that is like the main file, and then in this file, or like the root of the file, and then in this root you have a hierarchy of sub objects. You can have 3D arrays, matrices, you can have raster images with color palettes, you can have raster images without color palette, you can have just 2D arrays, you can have data sets as tables, and so on. It's very large things you can put in an HDF5 object, or like file. And you can access each one of the objects inside the HDF5 object or file by going through the hierarchy. So you have a path. So for example, if you want to access this matrix here, you will have to go to /foo/bar/temp, and then you will access this matrix only, and you can load just that object from the file and not load the entire file. So it's very convenient when you have like huge amount of data with many genes and you want to download only one from a huge matrix. You can download one row of one path only, and we can do that in TissUUmaps. I will show you a bit of an example now. Why did we want to add HDF5 in TissUUmaps? It's because it's like the de facto standard for single-cell RNA-seq and spatial omics. It's being used in 10x Cell Ranger software, so the software that really produces the data from raw files, and it's how they store the pre-processed data. It's also used in ScanPy, SquidPy, and all the scVerse library with AnnData. And the LUMPy format is also a format that is used in single cell and RNA-seq data, and it's also based on HDF5. So once we can read HDF5, we can read many different formats that are based on this. Even MATLAB in matrices are based on HDF5. And another big advantage is that it comes with a software library. So the HDF people, they develop interfaces to access the data in C, C++, Python, R, and then you have like some warper around these, like in JavaScript and so on. So we used one that is called H5WASM that can load files in JavaScript, and I will show you how that works. So let's take an example. Let's say we have this paper that we found online in a cell journal called Spatial Temporal Transcriptomics Atlas of Mouse Organogenesis using DNA Nanoball Patterned Arrays. And I took a random paper because they have a nice figure and with nice clusters in many samples. So I wanted to see if we could load that locally. And the nice thing is that they actually give all the data to download on their website. So if we go on their publication, we can find the link, and then we can download like all the mouse embryo old stage, and you see it's an H5AD, which means it's already an AnnData in H5 format. This is 7.2 gigabytes, but actually it's more like 20. So I already downloaded that, and I can show you how we can load that locally. So if we go in the AnnData examples here, we have the mouse embryo all stage.h5ad that we downloaded here. And now I will start TissUUmaps on the side. It's loading in the wrong window. So here it is. So now we have TissUUmaps here, and we have the file that we want to display. And I will show you how to load it just as a marker first. So if we want to load one object from this, we can go here and then go to the desktop where I put these TissUUmap 3.1 and AnnData example and mouse embryo. And now it's loaded. So even though the file is 21.6 gigabytes, you see that it loaded in less than a second because it only reads the main slash and then you can browse it. So if I want to say which coordinate are X, I can browse all of the objects inside of my HDF5. So if I go to observation, for example, here I have all the observation. If I want to have the x coordinate, they are stored in ops_m/spatial. We could also display the PCA or UMAP format, but let's say we want to see the spatial coordinates, then we can like display the row 0 and row 1 from spatial. And then we can directly update the view and it will load all of my coordinates directly here. So now we don't have any information about what it is, just X and Y coordinates. So we'll have to go to the render options to see what we want to display for each point. And again, we can select a key so that they will be sorted by a different key, and we can select annotations. And then we can generate random color for all the annotations. And directly we have the same annotations as in the paper. Unfortunately, here we don't have access to the palette that they used for having the same colors, because it's somewhere else in the AnnData format, but we can, just by loading this, load the main annotation in random colors. We could also decide to like have a different color space. Instead of grouping things per cluster, we could select another observation from the HDF5 image. And here, for example, select the total counts per point. So it's spatial transcriptomics. So each point in the data has each point has a number of counts in it. And if I select a color map, then I can display that. And here we have the color map. And then we can go through all the points. And we see that it's like for each point, we have a number of counts in it. So you can explore the data directly from what you just downloaded online and display it. But we can go a bit further because the format is already ordered in a way that we know it's actually an AnnData object. And AnnData format, it's an HDF5 file, but with very specific objects that we know about. So we have like the gene matrix in the middle, we have observation for each different things you have in the data, like clusters or number of counts and so on. You have the names of the genes in a different matrix and so on. So you have a lot of information that is structured in a way that you can actually load it always in the same way. So we added a pre-processing of these AnnData into TissUUmaps so that if you load an AnnData file with like open, then it will load all of the observation as drop down. So if I go here back to my TissUUmaps object, and instead of adding as markers, I directly load the whole H5AD in TissUUmaps, then it will like read the AnnData object, extract all of the observation. They can be numerical observation of categorical observation, and all of the genes that are in the X matrix. So here it's spatial transcriptomics. So we have more than 3,000 different genes, or like maybe a bit more, like 10,000. But we can like click on one, and then as it's HDF5, it will only load one row of the huge X matrix and then display it. So then you can see how it is. The nice thing also about that is that in AnnData format, you directly have the color map associated with each thing. So if I select the annotation here, you can see that I have the same colors as the colors they used in the paper. So you can directly interact with the data exactly as they did in the original paper. And you can like select one object here and say that this is type brain, and you can check everything that is brain in the data or some other things. So that's how to load AnnData. And then you can also share your view as a web page. So we said that TissUUmaps was based on a web browser, so you can always export to static web page, and I will show you later how to do that a bit more in detail. But just if you want to test, you can go to this web page where I uploaded this HDF5 image directly. So if I click here, it will load the TissUUmaps.scilab.lab.se web page, and you have access to all observations we discussed, and you can share that link with anybody that you would like. If it was our publication, we could also share the data as part of the publication, and people can go in and look at each point or select some part and export as CSV, or they can display like one gene and so on. So it's very flexible in what you can do with that. You could also edit the file to put what you would like in it. Now I wanted to show you how to load things in collection mode. And could do a small example. You can load things manually if you have multiple images. So if we go back here and close the file, we can go to a different folder. Here I have like 5 images and of DAPI and fluorescent channels, and I can load them into TissUUmaps. So if I drag and drop, then I can see my DAPI here, and in the image layers I have it here. And then you can take the fluorescent channels and put them on top. So now here you will have the different fluorescence channel and the DAPI, and you can zoom in and see the raw data. And right now they're on top of each other because we drag and drop just like as one stack of images. But here you have the collection mode. And if you put in collection mode, then you will have your 5 images next to each other. And then you could have markers that are on one specific item of your collection, or you could have one marker set with like a column that says on which image it should fit. So you can have like markers display on each one. So that's if you want to do it manually. If you already have an AnnData object and it comes with library ID, which is supported by like in SquidPy and so on, then you can load it into TissUUmaps, and then you have your different library IDs in collection mode. And here we can like select one at a time in the view, and we can see that here we have 3 samples, very similar but slightly different. We can add a bit of contrast maybe. Yes, here you can play with the different things so that you can see your images. And then if you want to select an observation from the AnnData, so if I go for like the initial annotations here, it will load annotations and with one on each library ID. So the annotations are connected to one library ID. If we go here and we remove the collection mode, then we will have all the markers on top of each other. And then you can see that they're actually not well aligned or anything because there are 3 different samples. But it's nice to see that in each sample here, the clustering or the annotation based on clustering are similar on the 3 samples. So it's very good example of like correct clustering. Okay, so that was about like the new feature, and the last thing I wanted to show you was how to integrate with Jupyter Notebook. So I created a small notebook example here. So if I load it in the browser, I hope it will work. Yes. And so first we import TissUUmaps, and we have a Jupyter module here, and you can import it as tmap. I also import some other packages I will use here just for simple testing. So now it's loading, and you can see which version of TissUUmaps you're loading. So here we have the last 3.1 version, and it also loaded the other modules. Then let's say we want to load an AnnData object. So we have one here that we looked at before. And then it will create a small window here where you will have your TissUUmaps instance. Very similar to what we had before. You have a way to interact with the data in the notebook. You can display clusters, you can play with it, and so on. You also can load it in a new window if you want to have it full screen, for example, so that you can see it bigger, or if you want to then work on the file and export it or anything else. So then you can load it in the main window, and in the same way you can interact with the data. So that's if you want to load an AnnData that is on the system, on your files. But we can also play with data from, for example, already existing datasets on SquidPy or on ScanPy. So if we use the SquidPy datasets objects or like package, we can load some of their examples. And here I will load a Visium, Fluo, and data file. So you load it, it will download the file and then put it in your memory. And then you can plot it with like ScanPy. And here you have your different clusters, different colors, and you can see it, but you cannot really do anything interactive with it. It's just like a view of the color you decided here. So then if we save that and open it with TissUUmaps instead, we can load that into the Jupyter notebook here. I call just Visium, Fluo, and data, and then you see you have directly your fluorescence background that was a bit hidden here. And you have your clusters with the same colors. And you can see it's very similar to what we had, except here you can actually zoom in and see more details interactively, or you could like display one at a time and so on. And then if you're very happy about how it looks in the view here of TissUUmaps, you can like crop it to what you would like, and then you can take screenshots. So if we take a screenshot of this viewer here, so I had the viewer here being TissUUmaps view, and then you have a screenshot that will be saved in your Jupyter notebook. Then if you want to display something else, like the total counts here, and you want to save that as a different screenshot, you can do that. Or if you want to have do especially on this, for example, you can like zoom on this part and then again take a screenshot. That's very convenient if you want to publish, or like if you want to put that directly online as a notebook, then you can display what you want from the TissUUmaps view. So now I wanted to talk about the network diagram graph, like graph visualization in TissUUmaps. And I took an example from the SquidPy documentation, where they actually used spatial neighbors to build a local connectivity of your data. And here I will use the grid coordinates. So I take this image and I will link all spots with a graph. So if I run these spatial neighbors here, then I will have a new observation in obsp.spatial connectivities here, and we can look at what it looks like. It's a 2800 by 2800 sparse matrix of floating points. And basically it's for each spot in the data, it says to which other spots it's connected. It also comes with spatial distances, so you know for each spot how far the others are. Right now we don't use that in TissUUmaps, but in the future we can we're working on having different colors depending on the distances and things like that. So if we use the spatial scatter that comes with SquidPy, we can display a subsample of the spots and see how they are connected. But as everything is stored in the H5AD here, we can also just write it to the disk and load it into TissUUmaps. As you can see, it has a small conversion from like matrix sparse to another format, but it's pretty fast. So here, if we display the clusters, now we can see that we have the spatial connectivity that is displayed directly, and we can play with like the size of each marker, and then the graph comes with it. We can hide the image and see just the graph and the points. We can also like go through, if I put that a bit bigger, yes, remove the background maybe. We can go through one cluster at a time and see how they are connected to each other and so on. And this is one example of connectivity, but you could have many more if we take like these non-grid datasets, so where you have the points that are not as a nice Visium grid. Then we can use a generic coordinate system in SquidPy. and load the spatial connectivity again and plot it. And then we see how it's connected. And again, we can load it. Oh, I run both. So now we have TissUUmaps that will run the cell type. And then you can see what it looks on the whole data with this kind of graph. And here again, we can see each cluster and where it fits on the network. And this is still a bit messy, so we could use a simpler graph representation. If we use like Delaunay and generic coordinate types, then we will have a plot that looks like this, where we have Delaunay triangulation. And then again, if we load it in TissUUmaps, we will have the Delaunay triangulation directly here. And you can see how neat it looks compared to this one. So here we have a Delaunay triangulation on top of your data, and you can play with that. You can also load different plugins and everything. So everything works as different as before, but now we also have the network in the background. And if you have a place where you're really happy about how it looks, you can always also take a screenshot with the network in the background. Then I can go to the last part I wanted to show, and that is how to export things to a static web page. As I showed you with the embryo, mouse embryo, you can access it online, but maybe so this was running on a TissUUmaps server, which means we have TissUUmaps Docker running on a Kubernetes server, but maybe you don't have that for your own use, and you only have like usual Apache server, like a web server. So then you can convert your images into a static page that can be loaded anywhere. So let's try an example with one of the files we had before. So if we take in the Jupyter notebook we were working with, in the AnnData objects, we have all the AnnData we saved. And then let's say we want to use the graph AnnData object here. I can open it in TissUUmaps. And here I will have the fluorescence background and all the measures that were computed in ScanPy, like the total counts of, total counts, like we can also display the log1p genes by count and so on, or the clusters, or the whole set of genes that are expressed in this tissue. And then I want to share that with someone online. So I will go to File, Export to Static web page. Then it will ask me to save it somewhere. So I can create a new static folder. And then inside of this static folder, I will export everything as one page that can be loaded on the server. It can take a couple of seconds, and then it will tell you when it's done. And then if we go here, we have the static folder. And inside this folder, we have the index webpage and some data folder and all the dependencies, JavaScript dependencies. So if we select that and we put that on the web server, then we would have a similar interface as the one I showed before. So we can test it here. If we go to this web server that is running locally here on this page, we have access to the static folder. And then you're in your browser and you have all the H5, the AnnData object that we played with before. And we can load the total counts and so on. Well, we should be able to. Let's check. We have to restart my server here. If you give me 1 second. Yes. So now if I go back to the server here, I directly have that, and now it's loading. And now you have your clusters. And you also have like the whole set list, gene expression list. and you can select one and display it. So you have access to everything. And if we look at what was produced in the data, you actually have your image background that is exported as a DCI file so that it can be loaded in the browser. And you have your H5AD file that is accessed as an HDF5 image or like object, and then it can load all of the observation from it. And then you can, yes, upload that on your server and share the link. You can also protect it with password, or you can put it for access to everybody else, to like collaborators, or if you publish your data in your analysis in a paper, in a journal paper, you can add the link as to access the data. And from the link, people will be able to download like any subsample they want, or the whole HDF5 if they want to. Any question on that export? If not, then I can move on to the conclusion, and that is a very like or global overall conclusion, but basically we developed TissUUmaps as a web-based interactive viewer, and you can load 2D images, regions, and markers either from separate files or from AnnData objects. We try to collaborate with all the other open source initiatives for cell and tissue segmentation and spatial statistics to have better compatibility with TissUUmaps. And if you have any questions or any problems running TissUUmaps, you can contact us. If it's a bug in the software, you can put an issue on GitHub. If it's a feature requirement or anything, you can also ask on the Image SC forum, where we are a collaborator to the forum. And then you can check all our galleries and other things on our website. So if you go to tissueumaps.github.io, here you have access to our gallery, and then you have many examples of what you can do with TissUUmaps and people who are using TissUUmaps. And then you have the whole documentation here, and you can check how to load different things in TissUUmaps. And yes, Frederick just put the GitHub page, so you can put so that's the main TissUUmaps GitHub, and you can put issues here, and we will try to fix that. Not so many questions so far, so we are good with time. We have lots of time if you want to ask questions now. So to answer Swan Liu, actually, so if you have just an Apache software server, then yes, you will need to export a static web page. So the question is, if I understand correctly, we have to export the single cell RNA and STSEC as static web pages separately. Yes. If you have a normal Apache server, then you would need to have each one as a different web page. If you deploy TissUUmaps as a Docker container, like we have on tissueumaps.scilabLab.sc, then you can just upload your TissUUmaps file and H5 image, and then it will create it directly as a web page. So you don't have to convert it. But you would need one for the single cell and one for the spatial transcriptomics separately, yes. If we go, for example, TissUUmaps is used in the Human Developmental Cell Atlas project. So if we go to their webpage, HDCA Sweden here, we have the list of organs. So if we select the lung, they have different techniques used. They have single cell, spatial transcriptomics, and so on. So if we select the spatial transcriptomics and one of the weeks that was sampled, then you have the cell types and you have a gene expression and so on. If we select single cell, and epithelial, then you would have like the cell types for epithelial cells, or if you want to display all of them, here it will display all the cell types for all your cells in your project. But you would have to work on having one of TissUUmaps instance for all the view you have. You need to create one for each. Here, this is running on our Scilab Lab Docker instance, so it's just putting the correct AnnData in the correct place. We also have in-situ sequencing data here. So if we select one of the in-situ sequencing and we display the mRNA reads, you can like zoom in and see the raw data in the background with all your targeted genes. And then you can select like one target at a time and display it and so on. You can also draw regions of interest and export data as CSV files. And yes, thank you for your interest, and thank you for joining the webinar. And you can stop sharing. And don't hesitate to contact us if you have any questions or things you want to try, and to try it yourself. So you can always go to our Jonas, are you? Oh no, you see that. You can yes. You can go to our webpage and install it locally and try it with your own data. Thanks. Have a nice day.
Thank you.
Thank you.